Uh, Thank you, ISMB, for this opportunity to present. So today I'm going to talk about interactive visualization of raw nanopore signal data with Squigualiser. The term Squigualiser comes from two terms, squiggles or signals for signals or squiggles and visualizer. So the combination makes it Squigualiser. So to give you an idea what nanopore data analysis is, it's a third generation sequencing technology. So you have different sizes of sequences. It essentially creates a raw signal, and that raw signal has to be base called using neural networks to get the FASTQ sequence. And once you have the signals and the FASTQ sequence, you can use some software to do signal level analysis. And in this kind of signal level analysis, one key stage is to actually map the signal back into the sequence. The sequence can be a read or a reference. So this is the alignment process. So you start with the raw signal, you do the base calling to get the FASTQ sequence. Once you have the FASTQ sequence or the read, you can map it back to the reference genome and get the read to reference alignment. In nanopore domain, you have two more alignments that you can do. One is signal to read and the signal to reference alignments. So even though we have many read aligners like Minimap2, Bowtie2, Barrow-Wheel aligner, they all adhere to the same standard of SIGA encoding as documented in SAM file format. Because of that, downstream analysis and making visualization software like IGV is very easy. But in signal domain, in nanopore domain, we don't have such a standard way to encode the signal alignment. So in our way to create a signal aligner visualizer, the first thing we wanted to tackle was come up with the encoding, which we named as signal string or shortly SS encoding. So what happens in SS encoding is it's actually again based on SIGA encoding. For example, if we have a SIGA encoding like one mapping, two deletions, again three mappings, two insertions, and one mapping, the SS encoding, for example, can be one mapping means it's just a base. So in the signal domain, what it means is there's a base that actually has 20 signal samples. And then we have two deletions. That means two deletions to the reference. That means you cannot find that part in the signal. So it's just going to be two bases deletion. Then you have three mapping. Three mapping means three bases mapped to the reference. So we in the signal alignment scenario, you have to have a single base resolution. That means it can be the first base having six sample points, followed by 7 sample points for the second base and 8 sample points for the third base. So the one insertion means there's an insertion to the reference. That means, again, just another base, which can be just 20 signal sample points. And then we have 1M, that means just another base, which can be just 3 sample points. So once we have this SS encoding, we were on our way to create a signal aligner visualizer. So these are like all the signal aligners available in nanopore sequencing domain. So you have a bunch of signal to read aligners and signal to reference aligners. So to make it easy for the nanopore community to use Squigualiser, we did the job of actually integrating our encoding method into the output of these signal aligners, which was a big task, but I think it was necessary to get Squigualiser out there for the nanopore community. Now going to the examples, I think everyone is familiar with IGV, and this is a read pileup where you are visualizing a variant, A and a T. And if these reads are coming from nanopore sequencing, which means that they originally have raw signals, now using Squigualiser, you can visualize them as a signal pileup. So you can see I have circled on the overlap up there that there's a clear difference between the current levels when there's a variant is present. So this tool has all the features of interactive visualization. You can zoom in, zoom out, you can drag, you can highlight, you can hide, and you can do various stuff that a normal interactive software would allow you to do. And this is one of my favorite examples, which I think for as a group who understands the power of visualization, this would have not been easy to detect if Squigualiser was not present. So here I have two data sets. One data set is methylated, where all the U's or the T's are methylated, and the other data set is like a controlled sample, where none of the bases are methylated. So we use a signal alignment method, which we call method A, you can see when the data set is non-methylated, it aligns the signals well to the sequence, so the reference. But when there's a methylation is present, you can see it actually include false deletions to the alignment. So this would have been really hard to detect if we don't have Squigualiser or any kind of visualization aspect to visualize the signals. So this, in contrast, this is method B, using the same data set, aligning the signals back into the reference for methylated and non-methylated case. You can see this method B is a robust method, which is not very sensitive for the methylations present. So these kind of things, I think, were possible after Squigualiser. Yeah, so Squigualiser is available as a free open source software on GitHub. You can give it a try. I would call it the IGV in signal space. And I would like to thank my supervisors, Ahira and Hasindu, and the amazing group of people that I work with at GTG Lab, at Garvan Medical Institute of Research in Australia, Sydney, Australia. So thank you very much. Open for questions now.
I have one quick question. Very nice talk. If you go a little bit back into your visual representation, yeah, I want to ask about the top row. If I understand correctly, this provides me an aggregated information from all rows together, right?
Yes, it's just an overlap of all the signals.
So that's what I wanted to ask, whether it's just pure overlap or whether you do something more interesting. Okay, yeah, sounds good. Thanks.
Thank you.